Okay, good day everyone. So now let's move on to a very interesting topic, okay, which is yung central dogma of molecular biology. So, napapahapayawan na natin to dati. So, palagi ko itong sinasabi sa inyo, central dogma, protein synthesis and all. So now, i-discuss na natin siya in, hindi naman sobrang detailed, pero on a deeper, okay, level compared dun sa dinidiscuss ko dati. So, head start lang. So again, yung central dogma of molecular biology natin is composed of basically three parts. So mag-start siya sa DNA natin. Okay, yung DNA natin, okay, will be replicated. So mare-replicate lang siya. So ito yung tinatawag natin as DNA replication. And then this DNA, okay, will be used to produce your mRNA or messenger ribonucleic acid. Okay, so anong process yung production ng mRNA from your DNA? It's the process of your transcription or the process of transcription. So basically, sa transcription, uh, it is a process of producing RNA molecule from your DNA molecule. So meron na tayong mRNA. And then this mRNA will be, okay, translated to produce your protein. Okay, so meron ka ng protein dyan. So anong process? It is the process of translation. Translation. So basically, ang nangyari lang dito, nire-read yung mRNA molecule natin to produce your protein. Ito yung reason kung bakit kailangan protektahan yung DNA. Kasi si DNA, okay, siya yung magbibigay ng information to produce your mRNA, and then si mRNA, siya yung magbibigay ng information to produce your protein. So kapag na-damage si DNA, madadamage din si protein. And then, as we all know, protein is very important sa atin kasi it is composed of mass. Basically, protein provides different functions sa atin. So, pwede siyang maging enzyme, pwede siyang maging structure, muscles, and all. So, again, ulitin lang natin, sa central dogma, composed siya ng DNA replication, transcription, and translation. So, ito yung overview ng central dogma of molecular biology. And okay, as we move on, ay discuss natin in detail yung per process dyan. So, yun lang. So, let's start. Okay, so now let's move on to our first topic, which is your DNA replication. So, basically, sa DNA replication, ang ginagawa niya lang is nire-replicate. Okay, from the name itself, nire-replicate niya lang yung DNA molecule natin. Or kung mas familiar kayo sa term na clone, kino-clone lang niya. Okay, so gumagawa ka lang ng another copy of your DNA molecule. So for example, we have this original DNA molecule. At the end of DNA replication, okay, magkakaroon ka ng dalawang DNA molecule. So from originally one, okay, magkakaroon ka ng dalawang DNA molecule. So bakit kailangan i-replicate or kailangan i-clone yung DNA natin? Ba't kailangan ng two copies? So remember, during your cell division, during your mitosis, specifically your mitosis, nag-share ng amount ng DNA molecule yung dalawang daughter cells na mapaproduce. So, di ba meron kang isang cell at the start of your what do you call this, cell division or mitosis, meron kang isang cell sa start, and sa end niya is meron kang dalawang cell. So, dapat, kung ano yung DNA molecule dito sa starting cell mo, dapat ganun din yung DNA molecule sa daughter cells mo na mapoproduce. Eh di ba sa part ng cell division natin, naghihiwala yung chromosomes natin. So basically, nandun yung DNA natin sa chromosomes natin. So pag naghiwalay ba sila, okay, mahati ba yung amount ng DNA natin? Okay? Yun yung question. Ang answer doon is hindi. Kasi nire-replicate natin yung DNA at the start of your cell division para pag naghiwalay siya, parehas pa rin ang set ng DNA from your original DNA molecule. So parang, for example, meron kang 23 chromosomes sa meron kang 23 chromosomes sa original cell mo. So at the start of your DNA replication, at the start of your cell division, dapat 46 siya. Para kapag nag-divide yung cell mo into 2, okay, meron kang tig 23 na, what do you call this, 23 na chromosomes at each daughter cells. So yun yung yun yung importance ng DNA replication para ma-conserve natin yung amount ng DNA molecule sa cells natin during cell division. Okay, so our DNA replication involves this following scenario or processes. So first, unwinding of the DNA. So nag-start yung unwinding of the DNA natin sa origin of replication. Ito yung sinasabi ko sa inyo previously sa sa lecture natin na yung origin of replication sa prokaryotes ay isa lang. Whereas dun sa origin of replication ng eukaryotes natin is multiple. So yun, dito nag-start yung replication natin sa unwinding of the DNA. So remember, your DNA is composed of two strands na naka-double helix. So i-unwind natin yon, then magbubukas siya. Dun yung origin of replication. And then after which, okay, after nang ma-unwind and mag-open nung dalawang strand natin, doon na mangyayari yung pairing ng bases in each strand. Okay, which is, as we all know, complementary dapat yung pairing natin. So, remember na yung adenine natin, okay, will always pair with your thymine. And then your guanine will always pair with your cytosine and vice versa. So, kung dito sa figure na to, okay, mapapansin nyo, okay, yung violet strand is your original DNA. So, natapos na siyang mag-unwind and nag-open na siya. Ngayon, ang ginagawa na dito is nagpe-pair na ng bases from the, what do you call this, from your original DNA to your new, to your original strand to your new strand. So, dito sa part na to, okay, so etong C, okay, nagpe-pair siya with your G. Okay, and then etong T, okay, from your original strand, okay, nagpa-partner siya with your A. So, ganyan lang yung process na yan. So, mag-open lang siya, then after niya mag-open, magdadagdag ng bagong strand. Okay, so after niya magdagdag ng bagong strand, so we will produce two new DNA strands, okay, that exactly duplicate the original DNA. So, basically, ito yung gist ng DNA replication natin. Okay, so hydrolysis energy. So energy from the hydrolysis of each nucleoside triphosphate. So as we build, okay, so this is your growing strand. This is your original strand. Okay, and then this is your growing strand. So as you can see, as we add, okay, base pair, okay, nagre-release tayo ng phosphate. Okay, so energy, okay, is released from the hydrolysis of each nucleoside triphosphate. So pag nagdaragdag ka, nagre-release ka lang ng pyrophosphate, pyrophosphate. So energy is used to form a phosphodiester bond to each nucleotide on the complementary strand. So, now, kapag kapag nagbi-build tayo, okay, ng bond, so nagre-release tayo ng nagre-release tayo ng pyrophosphate, but okay, we input, okay, energy to form the strand. Okay, so hindi lang basta magkakabit yan. Remember, we are building something here. So ibig sabihin, this process of replication is anabolic in nature, meaning, okay, we are building an expenditure of your energy. Okay, so yun lang. Remember, when we are building phosphodiester bond, so we use energy. Okay, ano yung phosphodiester bond? It is the bond between between your bases, okay, or nucleotides sa nucleic acid natin. So, asan ba yung magandang representation ng phosphodiester bond natin dyan? So, this is your nitrogenous base pair. This is your what do you call this? This is your ribose sugar, and then this is your phosphate group. And this bond between the phosphate group and the other sugar of another nucleotide, okay, this is the phosphodiester bond. Okay, ito yung phosphodiester bond natin. So this is the bond between the phosphate group and the what do you call this? The ribose group of another nucleoside. So kapag binibuild natin yon, kapag pinagdudugtong-dugtong natin yung nucleotides natin, okay, we use energy. Okay, so now let's discuss the direction of our replication. So first, let's discuss what is helicase. So basically, helicase is an enzyme that performs the unwinding or pagtatanggal ng buhol dun sa original DNA at several sections. So ibig sabihin, multiple yung origin of replication sa eukaryotes to. Okay, we're pertaining to eukaryotes. Okay, so at each open DNA section, okay, we call it as the replication fork. And then kapag open na yung DNA natin, yung original DNA natin or yung parent DNA natin, so here comes your DNA polymerase, which is which is also an enzyme which catalyzes the addition of nucleotide base pairs to your parent DNA, forming your new strand or your yes, your new strand. But okay, meron tayong what do you call this? Meron tayong kailangang rule na sundin. The rule is that your DNA polymerase can only add, okay, at the formation of 5 prime to 3 prime or at the direction of 5 prime to 3 prime. So ibig sabihin, nakakapag-add yung DNA polymerase natin sa direction na 5 prime to 3 prime. So ibig sabihin, 5 prime to 3 prime yun yung direction niya. So ibig sabihin yung strand na binubuo niya is ito rin. So 5 prime to 3 prime yung strand natin. So kapag tuloy-tuloy yung pagdadagdag ng DNA natin, okay, we call it as the leading strand. So kapag putol-putol, okay, yung pagdadagdag ng DNA natin, we call it as the lagging strand. And yung mga fragments na dinadagdag at a putol-putol na way or at a non-continuous way, we call it as the Okazaki fragment. So this, okay, later on, fragments will be joined, okay, using an enzyme known as ligase. Mas maintindihan nyo tong PowerPoint slide na to kapag nakita nyo yung, what do you call this, yung graphical representation ng DNA replication. Okay, so now here's the graphical representation of our DNA replication. So first, again, meron tayong helicase which unwinds your DNA. So, ito yung DNA natin. Okay, so ina-unwind siya and hinahati siya. Ina-expose natin yung both strands, okay, to form your new strand. So, kung papansinin nyo, okay, so these strands, okay, these two strands are your parent DNA. Maliwanag, this is your parent DNA. And then, as you can see, okay, yung direction nung nasa upper na parent DNA strand is 3 prime to. So most likely may 5 prime dyan. Okay, so ang direction ng upper strand is 5 prime to 3 prime. Okay? Ngayon, okay, so since your DNA can only since your DNA polymerase can only add at a 5 prime to 3 prime direction, so makapag-add lang siya from 5 prime to 3 prime. So yan lang. So habang nag-unwind yung DNA nyo, okay, dire-diretso lang yung pag-add nitong DNA polymerase natin dito. That's why it is the leading strand. So hindi putol-putol yung pagdadagdag ng DNA natin. But, okay, remember, sa kabilang pair, sa kabilang strand ng original DNA, kabaligtaran niya yung direction ng strand sa upper DNA. So, ibig sabihin, kung ito ay 5 prime to 3 prime, the lower strand is 3 prime to 5 prime. So, and, okay, kaya lang magdagdag ng DNA polymerase natin in the direction of 5 prime to 3 prime. So, ibig sabihin, habang nagbubukas to, okay, at habang nagbubukas dito sa part na to, yung DNA helicase natin, okay, paputol-putol niyang dinadagdagan yung strand na to. Okay, so that's why, okay, we call it as the lagging strand. Okay, so kasi naghihintay muna siyang magbukas yung DNA bago siya magdagdag. Unlike dun sa leading strand na habang nagbubukas yung DNA natin, dagdag lang siya ng dagdag. Okay, yun yung difference nila. So, in lagging strand, okay, the DNA polymerase is waiting for the original DNA molecule to open, okay, sa direction na pinag-oopenan niya bago siya makapagdagdag. Unlike sa leading strand wherein the DNA polymerase can readily add the nucleotides because it follows the direction of the unwinding of the DNA. Okay, so yun yung difference nila. And then we have here your Okazaki fragments. Ito yung sinabi kanina natin na paputol-putol na dinadagdag kasi hinihintay natin mabuksan muna yung DNA bago makapagdagdag. And then after, okay, kompleto na siya, okay, etong Okazaki fragments na to ay iko-completely seal or pagdudugtungin ng, okay, DNA ligase. So basically, ganun lang yung DNA replication natin. And kung papansinin nyo, okay, meron ka ng two new strands. So ito bagong strand to. Okay, and then ito ay bagong strand din. And okay, isang magandang i-take note, okay, the process of DNA replication is semi-conservative. Ibig sabihin, okay, dun sa mapoproduce mong new strands, okay, one part, okay, kalahati ng yung isang strand, okay, is from the original parent, okay, and the other strand is the newly synthesized strand. Kaya siya semi-conservative. Yung isang strand galing sa parent, yung isa bagong synthesize. So, ganun din dito sa isang to. This is the parent strand, and this one is your newly synthesized strand. So, that's why it is called as semi-conservative. Okay, now we're done with your DNA replication. So, basically, hindi naman detailed yung diniscuss ko sa inyo, pero ganun yung process ng DNA replication. So, nasabi ko naman sa inyo yung mga important parts like your DNA polymerase and all. Now, we can move on to another topic or another part of your central dogma, which is transcription. And very important sa process ng transcription is your RNA or ribonucleic acid. So, again, so this figure shows first your DNA replication, and then from your DNA, okay, you can produce your RNA, hindi lang mRNA, your RNA in general, through the process of transcription. Okay, so now let's discuss, okay, RNA or ribonucleic acid. So, napahapayawan na natin to before. So, main difference niya with your DNA is that it contains uracil, okay, a base pair instead of thymine that can be found in your DNA. So instead of thymine, we have your uracil here in RNA as your nucleotide base pair. Okay, so main function ni RNA is to provide or transmit information from your DNA to make protein. So specifically, okay, yung nagpa-function nun, okay, or nagko-contain ng information na yun is your messenger RNA or your mRNA. So it carries genetic information from DNA to the ribosome. So basically, you can produce your mRNA through the process of transcription or transcribing your DNA. Then we have your tRNA. Okay, so even though it does not directly contains the genetic information from your DNA, its main function also aids in producing or also helps in producing your or synthesizing your protein. So its main function is to bring amino acids to the ribosome to make the protein. So as you all remember, ribosome is the site for protein synthesis. So dito nangyayari yung protein synthesis, and yung nagdadala, okay, no specific amino acids papunta sa ribosome while reading your mRNA is your transfer RNA or your tRNA. Now moving on to your ribosomal RNA. So its main function is a structure. Its main function is to act as structural support, or it is tawag dito, it is a main component ng ribosomes natin. So si ribosome kasi natin, it's composed of both ribosomes, it is composed of both RNA and protein. So hindi natin mabubuo yung structure na ribosome or hindi tayo makagawa ng ribosome kung wala yung ribosomal RNA. So basically, it is very important in the process of synthesizing proteins. Okay, so just a recap. So mRNA, so it directly contains yung information from your DNA that will be used for producing proteins. And then the transfer RNA, from the word transfer, okay, it transfer amino acids papunta sa ribosomes while reading your mRNA. And then si ribosomal RNA, okay, from the word itself, okay, it is a main component, okay, to form or to produce your ribosome. So as we all know, main function of ribosome is to synthesize proteins. Okay, so here's another table showing the difference, okay, ng three different major types ng RNA. So, etong tatlong RNA lang yung i-discuss natin sa ngayon, pero as we go on or as you read about cell and molecular biology, we have different types of RNA pa. So, ito lang yung main RNA na ginagamit natin sa protein synthesis. So first we have your ribosomal RNA or RNA. So percentage of total RNA it is 80%, okay? And then its main function is a major component. It is a major component of your ribosome. So moving on, we have your messenger RNA or mRNA. So 5%, okay lang siya. And then it carries information for protein synthesis from the DNA in the nucleus to the ribosome. So, basically, yung kung ano yung information from your DNA, kokopyahin lang siya ni RNA, okay? Ni mRNA, specifically your mRNA. And then we have your transfer RNA or tRNA. So, its main function is to bring amino acids to the ribosome to the proteins. Okay, so later on, makikita natin yung process ng protein synthesis. Okay, now so let's discuss tRNA, okay, or transfer RNA. So, in your tRNA, okay, it has an anticodon which complements a codon on mRNA. So, ano ba yung codon or anticodon? So, remember, your mRNA is composed of sequence, okay? So, pwede siyang AUG, CCC, CGC. So, when you say codon, okay, it is basically tatlong magkakasunod na base pair sa sequence natin. So, for example, we start with AUG. So, this is a codon in your mRNA. And then we have CCC, okay, this is another codon. And we have your CGC, another codon. So, since nucleic acid pa rin yung mRNA natin, and your RNA, basically your RNA, we have what do you call this? Meron pa rin direction yung sequence natin. So for example, your AUG starts with 5 prime, okay? And then yung dulo nating dulong part na amino acid is your 3 prime. So kaya wag niyo kakalimutan yung basics ng nucleic acid natin kasi hanggang ngayon, okay, na i-apply natin yung 5 prime to 3 prime. So for example, this is the sequence of your mRNA. So 5 prime to 3 prime, AUG, CCC, CGC. So, alam na natin kung ano yung codon dyan. So, AUG, CCC, CGC. Ngayon, okay, in your tRNA, it will bring, okay, the amino acids which complements, okay, or, okay, it will pair, okay, dito sa later on na discuss, pero ang isipin nyo, since it has anticodon, okay, may part, okay, ng tRNA natin which complements or which will pair to your mRNA. So for example, you have your AUG here. So ang i-complement natin dyan is what? So UAC. So ito yung UAC. So ito yung sequence nung anticodon. So this is your codon and then this is your anticodon. And okay, kung papansin niyo, since your mRNA is 5 prime to 3 prime, yung direction dapat ng anticodon mo should be 3 prime to 5 prime. Okay? So, kung 5 prime to 3 prime yung kung 5 prime to 3 prime yung mRNA mo, okay, and it has codon, okay, yung anticodon should be 3 prime to 5 prime. Okay, maliwanag ba yan? So, for example, dito sa figure natin, okay, we have tatlong U, okay, as anticodon. And yung direction niya is, kung papansinin natin is from 3 prime, okay, to, papunta siya saan? 3 prime to 5 prime. So, ibig sabihin, okay, yung amino acid natin, which is the codon here, okay, should be 5 prime to 3 prime. And its sequence is A, tatlong A. So, this is your codon, okay, for your mRNA. So, it bonds to a specific amino acid at the acceptor end. So, meron tayong acceptor end dito. This is where your amino acid will attach. So, what do you call this? Hindi natin i-discuss yung process ng attachment ng amino acid to your tRNA, pero isipin nyo na lang si tRNA, nagko-contain siya ng specific amino acids. Okay, paano nyo nalalaman kung anong amino acid yun? So, nakadepende siya sa anticodon. Later on, we will discuss that. So, basta ang isipin nyo lang ngayon, your tRNA has anticodon, which complements your mRNA that contains the codon. So, yun lang yung gist na tong slide na to. Okay, in protein synthesis, two processes are involved. So, first we have your transcription, and then later on we have your translation. So, again and again, so transcription is just the process of forming your mRNA, okay, from a gene on a DNA strand. So, remember, kinukuha lang yung information from your DNA to mRNA. And one word na palagi natin inuulit-ulit na hindi pa natin formally na-define is our gene. So, when we say gene, okay, basically, ito lang yung part ng DNA natin which encodes for a specific protein. So, one gene, one specific protein. So, pag may gene ka, ang palaging isipin mo is meron kang may encode na protein. So, ganon. And then, we have your translation wherein your tRNA molecule, okay, brings amino acids to mRNA to build a protein. So, nangyayari to sa loob ng ribosomes natin. So, in your ribosomes, okay, it will read your mRNA, and while reading your mRNA, okay, your tRNA molecule will bring the amino acids needed for the synthesis of your specific protein. Okay, so your transcription is just the process of synthesizing or producing your mRNA or your RNA molecule. So in the case of here, we have your mRNA kasi meron tayong target gene. So first, a small portion or section of your DNA will be unwind, okay? So this small section of your DNA contains the gene. So maliit na portion lang siya ng buong DNA natin, and doon lang mag-unwind or doon lang magkakaroon ng site of transcription. And then, after nyo mag-unwind, so one strand, okay, is only be copied. So, isang strand lang yung gagamitin, okay, to copy or to produce your mRNA. Okay, so starting at the initiation point. So, meron naman tayo palagi na initiation point, whether it is, okay, in your transcription process. So, normally, ito yung mga nag-start to sa mga promoter region. Okay, basta portion siya ng DNA natin wherein it promotes the transcription of the gene. Okay, it follows. Normally, okay, in your what do you call this, in your eukaryotes, okay, it has a sequence, or yung initiation point na yun, it has a sequence of TA, or okay, natawag natin siya as TATA, okay, TATA region, or TA, yes, okay, so TATA, okay. It is in the case of your, what do you call this, in your eukaryotes, but in your prokaryotes, we call this region as Pribno box. Kaya siya tinawag na TATA kasi nga yun yung sequence niya, maraming TA base pair. And then an mRNA is synthesized using complementary base pairing. Okay, so for example, you have your DNA here. Okay, yung sequence ng DNA mo ay from 5 prime to 3 prime. Tapos meron ka, for example, na A, T, G, C, G, A. So, for example, ito yung sequence ng DNA mo. Okay, so isang strand lang yung i-copy, remember? So, ngayon, kapag expose na itong isang strand na to, okay, magkakaroon lang tayo ng complementary base pairing, okay, using your RNA polymerase. Okay, yung RNA polymerase, siya yung nagka-catalyze nung pagdadagdag or pag-base pair nung base pair. Okay, siya yung parang DNA polymerase sa replication ng DNA natin. So, RNA polymerase. So, ang gagawin niya lang, magdadagdag lang siya ng base pair complements to the DNA. So, RNA polymerase. So, enzyme din to. So, kapag open na tong strand na to, okay, or pag expose na tong strands na to, si RNA polymerase, magdadagdag lang siya, okay, ng base pair. Paano siya magdagdag ng base pair? From 5' to 3' kasi polymerase pa rin siya. So, una niya dadagdagan si A. Okay, si A magpa-pair na kay U. Okay, si G magpa-pair na siya kay C. Ganun pa rin, si C magpa-pair kay G, G, A, U. Okay, so now this color blue here is your RNA molecule. Okay, so ngayong itong newly formed na RNA molecule mo, okay, lalabas lang siya ng nucleus, okay, to be synthesized as protein. Ngayon, yung nag-unwind, okay, na DNA molecule natin, yung bumuka na DNA molecule natin, babalik lang siya on its original conformation. So, parang ano lang siya, nagnakaw lang siya ng information, then lumabas na ulit yung mRNA molecule natin. So, basically, that's the process of your transcription. So, somehow, it is almost the same sa DNA replication. Ang difference nito is isang strand lang yung kinokopyahan ng information, and then after makopyahan ng information, aalis na. And another thing, a small portion lang ng DNA natin yung kinokopya, which encodes for the gene, okay? And then yung nagka-catalyze nung pagdadagdag or pangongopya nung base pair, okay? Nung pangongopya ng information is your RNA polymerase. Okay, yun yung tandaan nyo. Okay, so here's a graphical representation of your replication. So overview lang din to. Hindi ito yung exact detail niya, pero at least, okay, alam niyo yung basic na sequences or basic na processes or steps na nangyayari sa transcription natin. So, again, as I've said earlier, okay, nangyayari lang siya sa a certain portion ng DNA natin wherein yung certain portion na yon contains your gene. And after nyong magbukas, okay, helicase din yung gamit dyan. After mag-open ng DNA natin, only one strand will serve as the source of information to form your RNA transcript. So in the case of this figure, okay, yung 3' to 5' strand natin yung nag-serve as the template strand or okay, antisense strand. So pag sinabi natin antisense strand, dito manggagaling yung information, okay, na kukuhanin ng RNA transcript natin. And when we say sense strand, basically, same yung same yung sequence niya with your RNA transcript kung papansinin niyo, except that instead of thymine, nagiging uracil siya sa RNA transcript. So pag sense strand, same yung sequence niya with the RNA transcript, papalitan niyo lang yung thymine into uracil. Okay, so and kung makita nyo dito, okay, gumagamit tayo ng RNA polymerase to form your RNA transcript. And again, kung 3' to 5' to, what do you call this? Yung direction ng production ng RNA transcript natin will always be from 5' to 3'. So, tandaan nyo palagi yan, basta polymerase, ang kaya niya lang gumawa ng transcript or strand from 5' to 3' direction. So, tuloy-tuloy lang yan hanggang mabuo yung mRNA transcript natin. And after niyang mabuo, okay, aalis lang siya ng nucleus, and then itong nagbukas na part ng DNA natin, babalik lang siya sa normal state niya. So, ngayon meron ka ng RNA transcript or mRNA. So, basically, ganun lang yung transcription natin. Okay, so it's medyo late. Oh, it's a bit late. Pero let's discuss RNA polymerase. So, nasabi ko naman na kanina kung ano yung main function ni RNA polymerase. So, basically, para lang din siyang DNA polymerase. So, your RNA polymerase moves along your DNA template, again in the 3' to 5' direction to synthesize your, what do you call this, your corresponding mRNA. So, let's try to decipher this one. So, okay, so from 5 prime, huwag kayong malito ba? So, ang sinasabi ko kanina is from 5 prime to 3 prime na direction. So, ang sinasabi niya dito is in the 3 to 5 prime direction. I think that's a bit confusing kasi yung palitan nyo to. So, from 5 prime to 3 prime direction siya. At to synthesize yung corresponding mRNA. So, so after niyang synthesize, okay, your mRNA will be released at the termination point. Okay, so yun lang siya. And then kung papansin niyo dito, we have your non-template strand and then we have your template strand here. So yung template strand, siya lang din yung what is antisense strand, okay, wherein your the information of your mRNA will come from. And then yung non-template strand, basically siya yung sense strand wherein its sequence is almost the same as your mRNA molecule. So again, so kung 3' to 5' to, and then this one is 5' to 3'. So, yung movement niya is from 5' to 3' na direction. Okay, so, yun. Okay, so ituloy lang natin yung kwento. So, ah kapag nakapag-produce ka na ng mRNA molecule, okay, so magde-detach lang siya sa DNA natin and it will leave your nucleus. So, mag-attach na siya sa ribosomes natin for translation. Pero, hindi ganun kasimple yung what do you call this, yung mga nangyayari. So, before pa siyang ma-translate into a protein, nagkakaroon tayo ng what do you call this, mRNA processing. So, we will have a different, we have a separate na discussion about mRNA processing, which I will discuss later on. Pero, for now, ang intindihin nyo, okay, is kapag natapos yung transcription from your DNA, nakapag-produce ka ng RNA script, lalabas siya ng nucleus. Pag lalabas siya ng nucleus or bago siya lumabas ng nucleus, nagkakaroon ng processing dun sa RNA transcript na yun before maging fully committed na mRNA molecule siya. So, in the next slide, I will discuss processing or pre-mRNA processing na nangyayari before matranslate yung mRNA natin. Okay, so now we have your mRNA processing as I've said earlier. Okay, now we have two new new what do you call this? New nouns. Okay, we have your exons and introns. We have two new words. So kapag sinabi natin exons, okay, they are the regions which codes for proteins. And when we say introns, okay, they are regions in your DNA that do not code for proteins. Okay, so para mas madali niyong matandaan, okay, si introns ay intervening sequences. So, ibig sabihin, nag intervene siya. So, wala siyang kwenta. So, dapat siyang tanggalin. And then si exons, siya yung mahalagang part ng DNA natin kasi sa kanya manggagaling yung information to code your or to produce your proteins. Okay, so a pre-RNA is formed that includes non-coding introns. So sinabi ko ito kanina, okay, pag na-transcribe na yung DNA nyo, okay, it both contains, okay, exon and introns. And para maging fully committed yung mRNA natin to be synthesized, kailangan muna tanggalin yung intron. So, natatanggal natin yun through splicing. Okay? So, basically, tinatanggal lang natin yung introns and iiwanan natin yung exons. Okay? So, the non-coding introns are removed. Okay? And then, after nilang ma-remove, okay, yung exons natin, pagkasama-samahin lang yan to form your mRNA. And then, dun pa lang siya mapaprocess to form your protein. So, we have a separate discussion dito sa mRNA processing na to. maybe next topic sa gene regulation natin, wherein i-discuss natin fully, okay, kung ano yung ginagamit ng cells natin to regulate yung production ng protein. So, mag-stay put lang kayo, basta tandaan nyo, introns hindi kailangan, exons kailangan para makapag-produce ng proteins. Okay, so here's a graphical representation para mas ma-imagine nyo. So, this two is your DNA. So, we have your non-template strand and this is your DNA template strand. And then, it will undergo transcription to produce your pre-mRNA. So, meron ka ng RNA molecule. And then, this pre-mRNA molecule contains both exon and intron. So, meron dyan exon, intron, exon, intron, exon, intron. And then this, okay, pre-mRNA will be processed para matanggal yung mga introns natin. So, dapat tanggalin yung mga introns, leaving your exon. So, kapag meron ka ng mRNA or RNA molecule na puro exons na lang, this molecule is ready to be translated to produce your proteins. Okay, now so let's move on to genetic code. So we've tackled transcription. So this topic, genetic code, is very important para maintindihan nyo yung process ng translation, para malaman nyo kung paano na-translate yung codes from your mRNA into sequence ng protein. So madali lang to, para lang siyang pang-elementary na topic kasi titingnan mo lang yung letters tapos meron na siyang corresponding na amino acid. So later on, mapapakita ko naman sa inyo yun. So huwag kayong masyadong kabahan. Madali lang to, pang elementary lang tong topic na to. So onting logic lang ang gagamitin niyo dito and then you're good to go. Okay, so kaya ko siya sinabing madali kasi nag-exist yung tinatawag natin na genetic code. So basically, your genetic code contains yung sequences ng nitrogenous bases natin in your mRNA, which, okay, determines kung anong amino acid yung nagka-correspond for that nitrogenous base. So basically, these nitrogenous bases are read in three bases or triplets or yung tinatawag natin as codon. So, napapayawan na natin yung codons in the topic earlier. So, basically yung codons natin, base pair lang siya in three. So, for example, AUG. Okay, this is a codon that can be found in your mRNA. And this AUG, okay, as I've said earlier, correspond to a specific amino acid. Okay, so it has different codon for all the 20 amino acids. So, di ba ito yung diniscuss natin in your proteins and amino acids. So, meron tayong 20 naturally occurring amino acids. So, every amino acid, meron siyang corresponding codon, which is needed, of course, to build your proteins. Okay, so your genetic code contains codons that signal the start, okay, and end of the polypeptide chain. So, yung start natin, madali siyang tandaan kasi ang codon na to, okay, or yung sequence ng mRNA dito is palaging A, U, G. So, ibig sabihin, kahit may mga sequences dito na C, 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 G, okay, dapat ang hanapin yung codon is palaging AUG. Pag nakita nyo yung AUG, ibig sabihin, start na ng pagta-translate. And okay, yung AUG, okay, nag-iisang amino acid lang yan, okay, which is your met or methionine. And then si end, okay, meron tayong tatlong codon dito, your wa, uga, huwag. Okay, so later on, ipapakita ko sa inyo kung anong itsura ng genetic code. Okay, so here's your genetic code. So huwag kayong mag-alala, hindi nyo kailangang i-memorize to kasi sa lahat na exam or quizzes na makikita nyo related with your genetic code, given tong table na to. So as I've said earlier, nire-read or binabasa natin yung mRNA natin into 3 or your codons. So ibig sabihin lahat yan, tatlo. So UUU, UUC, UAAN, so on and so forth. But okay, ang gusto kong i-note dito is that yung start palagi natin will be your AUG. Okay, which corresponds to methionine, which is an amino acid. And okay, itong tatlong stop codon which okay, nagsisignal siya na dapat i-stop mo na yung pagbabasa ng mRNA. So pag nakita mo to, stop na. Kahit meron pang kasunod, basta nakita mo tong tatlong to, mag-stop ka na ng pagbabasa ng mRNA. Okay, so we have your wa, okay, huwag, and uga. So, meron, para madali niyong matandaan, meron ditong palatandaan. So, para ka lang nag-aalaga ng bata, 'di ba? So, ang sasabihin mo, huwag, okay? Huwag ugain, okay, yung bata kasi iiyak siya, huwa. So, ganun lang siya kadali. Huwag ugain yung bata kasi iiyak siya. So, and then, yun, okay. So, tawag dito, meron tayong mga codons na kahit different sila, okay, nagko-code sila for a specific amino acid. Pero kung papansinin nyo, okay, kadalasan ng codons natin, magkaparehas yung unang dalawang letters and dun lang sa dulo nagkakapalit. So, I'm not sure kung i-discuss natin yung mutation here, pero medyo mahalaga tong dulong part na to kasi okay, minsan nagkakaroon ng mutation sa DNA natin so naiiba yung sequence ng amino acid, sequence ng nitrogenous base. So minsan, may natawag tayong mga silent mutation wherein kahit may iba yung sequence nung DNA natin, walang nagbabago sa sequence ng protein natin. Kasi nga, as you can see, kahit mabago tong U into C, still it corresponds to leucine na amino acid. So we call this as silent mutation kasi walang nagbabago sa sequence ng amino acid natin. Okay? So, kaya ko rin sinabi na parang pang-elementary lang to or pang-kinder kasi basically titingnan mo lang UCU, okay? UCU. Okay, nagko-correspond siya for serine. So ilagay mo lang doon serine. And then if yung kasunod niya na code dun is CCU, okay, ilagay mo lang doon as proline. So parang ganun lang siya, parang simple logic lang yung gagamitin niyo dito. So I hope later on matuto kayo kung paano mag-translate, okay? Okay, so let's try this one. So for example, you have this sequence of your amino acid, sequence of your mRNA. Remember, since nucleic acid siya, meron pa rin direction yung strand natin. So this one is from 5' to 3'. So from 5' to 3'. So ang gusto kong i-note dito is that yung mRNA natin, palagi siyang binabasa from 5' to 3'. Palagi nyo hahanapin kung nasan yung 5', then dun kayo mag-start magbasa. Okay? So, dito, madali lang siya kasi nag-start na agad tayo sa 5' to 3' na direction. So, we have your 5' to 3' here. So, agad, okay, nakita natin yung AUG. So, AUG, okay, it is the start codon. So, and then after that, so babasahin lang natin si CU, okay, then AGC, okay, GGA, and CUU. And then, okay, pupunta tayo sa genetic code. And then, if you will look to your genetic code, AUG, okay, codes for meth. Okay, so or methionine. And then CCU codes for proline or pro. And then AGC, okay, codes for serine. And then GGA codes for glycine. And then CUU, okay, codes for leucine. So, uh, basically, ang magiging sequence ng amino acid natin is that it will start into a methionine amino acid, okay, followed by proline, serine, glycine, and then leucine. So, from your mRNA, okay, it will be read or it will be translated into a protein sequence or amino acid sequence. So, kung may dalagdag pa dyan, for example, nakita mo na yung huwag, okay, UAG, ibig sabihin, stop ka na ng pagbabasa ng mag-stop ka na ng pagbabasa ng mRNA. And then, katulad ng sinabi ko kanina, nagbabasa tayo from 5' to 3'. Paano, pag binigay sa inyong, what do you call this? Paano, kapag ang binigay sa inyong sequence ng mRNA is from 3' to 5'. For example, meron kang A, C, 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 U, C, G, G, U, A, C, C. So, ang gagawin mo, babasahin mo siya palagi from 5' to 3'. So, kahit naka 3' to 5' to, mag-start ka palagi sa 5' na start. Okay? So, 5'. Oh, C, C. So, hindi siya hindi pa siya pwede. CCA, hindi siya start codon. CAU, hindi siya start codon. AUG, okay, this one is start codon. So dito ka mag-start magbasa ng what do you call this? Amino. Dito ka mag-start magbasa ng mRNA. So AUG, CUC, CCA. So AUG, CUC, CCA, itong tatlo na to yung codons mo. Okay, so pabaliktad mo rin siyang babasahin. Okay, so ganun lang kadali. Basta ang palagi niyong ilagay sa utak nyo, mag-start ka sa 5 prime. And then, ang palagi mo hahanapin after that is yung AUG. So, 5 prime and then yung AUG. And then, kapag naka-encounter ka na ng stop codon, di mag-stop ka na ng pagbabasa ng amino acid. Ganun lang siya kadali. Okay? Okay, so now let's move on to the translation proper. So, again, it starts with, what do you call this? The activation of your tRNA using your tRNA synthetase. So later on, i-discuss naman na natin ito. Okay, so now let's start with your tRNA activation. So remember, after ma-process yung mRNA natin, kapag fully committed na siya, okay, lalabas siya sa nucleus and then nasa cytoplasm na siya. And then it follows by the activation of your tRNA. So this activation occurs when your synthetase, okay, uses energy in the form of your ATP to attach, okay, or to condense a specific amino acid to your tRNA. So your tRNA contains an triplet of base pair, or meron siyang anticodon which complements your codon in mRNA. Okay, so ito yung anticodon natin. And then, bakit kailangan ng anticodon? Okay, this ensures na tama yung ilalagay natin na amino acid to your what do you call this, to your mRNA. Kasi kapag mali, okay, yung anticodon natin or nagkaroon ng mali sa anticodon natin or mali sa codon natin, okay, for sure maapektuhan din yung sequence ng amino acid natin. And then, so again, ulitin lang natin, ang function ni tRNA is to bring amino acid to your ribosome for the production or for the synthesis of protein. So, yun. Okay, yun lang naman yung sinasabi ng slide na to. Okay, so now let's start with the events that happen during your protein synthesis. So first, we have your initiation. And then after that, we have your translocation. And then after that, we have your termination. So first, let's discuss kung ano yung mga nangyayari or mga events, okay, that happen in your initiation. So first, okay, your mRNA attaches to a ribosome. Okay, so this is a simplified event. Okay, wag na natin intindihin yung assembly ng ribosome and yung mga factor needed for the attachment of mRNA. Basta ang isipin nyo, okay, your mRNA is attached with a ribosome. Ganun lang siya ka simple. And then, after that, okay, hahanapin ng mRNA natin, hahanapin sa mRNA natin yung start codon. Okay, nasabi ko na to kanina. So, yung AUG na sequence. And then, si AUG, very unique siya kasi sure na methionine yung tRNA na, sure na yung tRNA na maghanap sa kanya contains a methionine na amino acid. So, after mag-attach ni mRNA sa ribosome, hahanapin yung AUG. And then yung AUG codes for a methionine na amino acid. And then after that, okay, yung second codon attaches to a tRNA with the next amino acid. So basically, binabasa na yung what do you call this? Binabasa na yung mRNA natin. And then, for example, nakita na natin na ganitong code or ganitong codon, yung kasunod, okay, a peptide bond will form between the adjacent amino acid. So, again, dinadala lang kasi individually ng tRNA natin yung mga amino acid. And then remember na yung amino acid natin should be joined para makabuo tayo ng what do you call this, ng isang protein. So, kapag individual lang siya, nonsense. So, dapat ipag-join siya or ipag-bond siya with each other in the form of peptide bond. So, yung peptide bond, this is the bond between your amino acids. Ganun lang siya. Okay? And then, nangyayari siya during your initiation process, yung formation ng peptide bond between your adjacent amino acid. Okay, so yun lang. Okay, so now let's move on to another event, okay, which is your translocation. So after nang mabuo yung bond between the first amino acid and then the second amino acid, okay, kung anong amino acid man siya, okay, the first tRNA detaches from the ribosome. So self-explanatory, umaalis lang siya sa ribosome. And the ribosome, okay, shifts to the adjacent codon on the mRNA. So pupunta lang yung ribosome sa kasunod na codon ng mRNA. And then babasahin niya yon. After niyang mabasa, okay, for example, yung codon na yon, nabasa niya na. So may darating na bagong tRNA or amino acid which attach to the binding site of your ribosome. And then after niyang makapag-attach doon, okay, dala-dala niya na yung amino acid. And then after niyang, okay, idala yung amino acid, another peptide bond will form and empty the tRNA that, and the empty tRNA detaches. So aalis lang yung tRNA na nagdala nung amino acid natin. And then basically, okay, si translocation natin, siya yung parang elongation process kung saan dun na humahaba or dun na pinapadami yung amino acids natin to form your protein. So, first we have your initiation wherein nag-initiate lang, okay? Dumarating lang yung first amino acid and then dumarating yung second amino acid. Wherein sa translocation, dito na natin pinapahaba, okay, yung sequence ng amino acid natin to form your protein until, okay, ma-reach niya yung stop codon which we will discuss later on. Okay, so here's a graphical representation of our translation. So, wag tayong ma-overwhelm. So, let's digest kung ano yun nandito sa figure na to. So, this blue strand is your mRNA, okay? And then in your mRNA, makikita natin yung mga sequence or mga nitrogenous base. So, gets pa natin yan. And since yung movement ng amino acid natin, ang movement ng ribosome natin is into this, okay, direction. So, i-assume natin na from this dulong part ng mRNA is your 5 prime and then dito sa dulong part is your 3 prime. Okay, so from 5 prime to 3 prime. And then this, okay, this color violet, okay, itong color violet na to, and then this color blue is your ribosome. So, what I want you to take note here is that we have two subunits ng ribosome natin. We have your large ribosomal subunit, this one, and then we have your small ribosomal subunit. Merong specific function kung bakit may ganyan dyan, but may large and small, but mag-focus na lang tayo sa main function ng ribosomes natin which is to read and synthesize yung protein natin. And then this, okay, yung parang clip na to, itong mga clip na to, okay, is your tRNA. Yung mga clip na yan, yan yung tRNA natin. And itong mga bilog-bilog na to is your growing protein sequence or your growing peptide chain. As you can see, okay, nandito na tayo sa elongation stage or elongation or translocation stage wherein, okay, dinadala, okay, ng tRNA yung amino acid based from the codon. So, meron tayong AUA here and then ang specific na amino acid that corresponds to your AUA is your ILE or isoleucine. So, pagkadala niya dyan, okay, a peptide bond will form between your isoleucine and then your growing peptide chain. And then kapag, okay, nag-bond na to, yung alanine and your isoleucine, may empty na ngayon yung tRNA natin kasi wala na siyang laman na amino acid. Kapag na-empty na siya, okay, i-release lang siya from your ribosome. So kapag na-empty na siya, okay, ready na ulit pumasok yung bagong tRNA to bring kung anong amino acid yung kailangan based from the codon na kasunod. So, GUC. Okay, so yun yung next na codon natin. So, darating yung specific na amino acid na nagko-code for your GUC. So, ganun lang yung basically elongation or translocation stage. So, pinapahaba na lang natin yung growing peptide chain. And then, what I want you to note here is that yung bonds between your amino acids is what we call as, what do you call this? Peptide bond. So, yun yung bond between your amino acids. So, peptide bond. Okay, now so pupunta tayo sa last event during our translation process, which is termination of protein synthesis. So, this is very self-explanatory kasi termination meaning ini-stop na natin yung process. Okay, so tina-terminate na natin yung process ng protein synthesis. Okay, so this termination occurs okay when all the amino acids for a protein is synthesized. Ibig sabihin, kumpleto na yung sequence ng amino acid natin. And then, okay, nangyayari to, okay, when our ribosome, okay, reach a stop codon, okay, yung uga, hua, and uwag. This is very, okay, what do you call this? Medyo delikado rin to, okay, so minsan, okay, kapag merong mutation na nangyayari sa DNA natin, minsan, prematurely na stop yung pagsisynthesize ng amino acid. So, ibig sabihin, for example, kalagitnaan pa lang ng polypeptide chain natin, okay, sinisynthesize siya, biglang nagkaroon ng mutation na iba yung sequence, okay? Biglang naging stop codon yun. So, ibig sabihin, mas mataterminate na agad prematurely yung pagsisynthesize ng protein natin, which is very dangerous. Lalo na kapag yung protein na sinisynthesize or, okay, yes, kapag yung protein na sinisynthesize natin is very crucial sa katawan natin, okay? And then there is no tRNA with anticodon for stop codon. So walang tRNA doon, so walang amino acid na magdadala. And then after ma-terminate siya, okay, your polypeptide detaches from the ribosome. So umaalis lang yung polypeptide chain natin or yung protein natin to our ribosome. Okay, so to summarize, yung protein synthesis natin, okay, so first, an mRNA, okay, attaches to a ribosome. Okay, and then hanapin yung AUG doon and then ilalagay yung methionine. So molecules of tRNA bonded to a specific amino acid attached to the codons of mRNA. And then after nilang mag-attach dun sa codons natin sa mRNA, okay, a peptide bond will form between the growing peptide chain and your amino acid na ka-attach pa lang. And then, after maka-encounter ng ribosome natin ng stop codon, okay, when our polypeptide chain is complete, okay, mag-stop lang or mata-terminate lang yung protein synthesis. And after ma-terminate yung protein synthesis natin, okay, your polypeptide chain will detach from your ribosome, okay? And nakalagay dito as to function as an active protein, okay? Minsan, okay, readily nagagamit na agad yung protein na yan, pero minsan meron tayong tinatawag na post-translational modification wherein yung protein natin minomodify pa siya. Nilalagyan ng mga, for example, lipoprotein siya, lalagyan siya ng lipids. For example, glycoprotein siya, lalagyan pa siya ng carbohydrate. So, meron tayong tinatawag na post-translational modification wherein minomodify lang natin after translation yung protein na na-synthesize natin. So, meron tayong separate topic for this one. So, di-discuss natin yun later on sa subject na to. Okay, so this is an overview or graphical representation ng protein synthesis natin. So, after transcription, okay, from your DNA, after transcription here, from your DNA, from your nucleus, okay, lalabas yung mRNA natin. And then after niya lumabas, okay, mag-a-attach siya to a ribosome. And then following that, okay, meron tayong activation ng tRNA natin, okay, using your tRNA synthetase. So basically, ang ginagawa ni tRNA synthetase, kinakabitan niya ng amino acid yung tRNA molecule natin. So after niyang makabitan, okay, pupunta na siya dun sa ribosome natin kung kailangan siya. Kapag nabasa ng ribosome natin na, for example, itong codon na to corresponds for this tRNA, so pupunta siya dito sa ribosome na to. And then after niya makapunta dyan, yung daladala niyang amino acid, Okay, and then the growing peptide chain will form a peptide bond. And then after maka-encounter ng stop codon ng ribosome natin, mag-stop lang yung protein synthesis. And then, after mag-stop yung protein synthesis, magde-detach lang yung protein natin from our ribosome. And then, if kailangan ng modification, so mamamodify lang yung protein natin, pero pag wala nang kailangan modification, okay, okay, magiging functional na yung protein natin. So, basically, yun yung overview ng central dogma of molecular biology natin. So, ah hindi siya gano'n ka-detailed as compared kapag mag-deep pa tayo kasi tinanggal natin yung mga regulators there. Pero I hope maintindihan nyo, okay, kahit pa paano yung sinasabi ko before dun sa mga previous discussion natin kung ano yung central dogma of molecular biology. So I hope na-enjoy nyo to. This is a very interesting topic kasi etong central dogma of molecular biology is the foundation kung bakit na kapag RT-PCR tayo. So, very familiar kayo sa RT-PCR kasi di ba yun yung ginagamit natin ngayon sa pagdedetect ng COVID, di ba? So, meron tayong tinatawag na PCR, which is your polymerase chain reaction, wherein yung concept behind that PCR is very or similar or hindi natin sabihin. Parehas na ang DNA replication. So basically, nagre-replicate tayo ng DNA sa laboratory. So, ang foundation nun, of course, your central dogma of molecular biology. So, three steps lang siya: DNA replication, transcription, and translation. Pero, sobrang daming nagawa, okay, na study dyan. So, basically, this central dogma is a course sa UP Diliman, which is your molecular bi sorry, BS Molecular Biology and Biotechnology. So, this, okay, topic na to, isipin nyo, isa siyang course or isa siyang degree sa UP Diliman. So, kaya very interesting tong topic na to. So, I hope you enjoyed our discussion. So, yun lang. Have a good day.